started while everyone else comes in. Next up is Nenad Pavin. If you have a question, raise your hand and one of us will come running to you with a mic. Ask after that. Thank you.
Okay, first I would like to, can you hear me actually? Is it working? Yeah. Oh, okay, perfect. So first I would like to thank the organizers for organizing this great meeting, and it was really a pleasure here to give a talk. Eva already mentioned in the previous talk what happens when the cells are dividing, and she also explained that when cells are dividing, they can actually properly segregate chromosomes or divide it incorrectly. But her talk was mainly focused on what happens during one cell division. Here, I will try to explain what happens with the chromosomes when, or with the cells when multiple cell divisions take place. So, again, something what you already have seen in previous talk, cells can get correctly divided, where the cells are dividing the same number of chromosomes between two daughter cells. In this simple example, you can see 3 chromosomes going to one daughter cell, 3 chromosomes going to the other daughter cell, and this is how the correct division looks like. On the other hand, sometimes missegregation takes place, and daughter cells are ending with an unequal number of chromosomes, and in this case, these cells are not happy anymore, they are not healthy, and this type of process occurs in different bad situations like miscarriages, tumor tissues, and the other in the other diseases. This is what happens during the cell division. But now Eva already has shown what the chromosome content look like, and here is the chromosome content of one healthy. I'm emphasizing this healthy diploid cells, where in the humans we have 46 chromosomes that are actually represented with 23 different chromosomes, each appearing in 2 copies. In the mathematical description that we are using, we are taking into account that each chromosome can have a certain number of copies, and it is represented by this vector, where each copy number here, for instance, chromosome 1, has 2 copies, and these 2 copies are represented by the number 2, and so on. And this is the case of this healthy cell. Of course, during this incorrect cell division, these aneuploid cells, the cells that do not have the proper number of chromosomes, getting very diverse. What means diverse? For instance, here you can see that chromosome 1 has 3 copies, chromosome 2 have 3 copies, 3 have 0 copies, 4 have 2 copies, and so on. And in this case, you can see that the mathematical representation of the situation here is with this vector that is pretty much diverse. But it's unclear what does this mean to us and how this is really diverse, and which type of problems this brings. So if you want to do some modeling, first we would like to understand with how big system we are dealing here. And for instance, I just wanted to calculate how many of different combinations that can appear in the system. Sorry, is this mouse pointer? Okay. How many different combinations can appear in the system where a certain chromosome copy number can vary from 1 to 5. And in the case of 23 chromosomes, it's very simple mathematics, 5 to the power of 23, and we are now dealing with quadrillions of combinations. This is extremely big number of combinations, and for me, a theoretician, this sends me also number of possible free parameters. For experimentalists, means that you will need like quadrillions of students to analyze all these karyotypes. So it's not, it's not doable. Only what we can do here, we can try to narrow down the number of unknowns. additional problem to this is we are not dealing with a small number of cells. For instance, this is number of cells in human body. We also need, if you want to calculate, we need to take into account that we are dealing with a really big number of cells. And the question is now how to narrow down unknowns here. It's not ideal situation, as I explained. Systematically, one would really need to do this. What we are trying to do is actually we are trying to identify what are the key processes in this tumor development or development of tumor karyotypes. So we are searching for some appropriate computational approach, and then to combine this and search for explanation in experimental studies on tumor karyotype evolution. So first, we wanted to somehow get to this point by a theoretical approach where we wanted to have information, all important information about certain karyotype, but to reduce this space in order to make it possible to calculate. I maybe should also mention that these trillions I mentioned before, that even one cannot calculate in computers because there is no computer with such big memory. So we even from computational point of view, we need to reduce the dimensionality. And what is our idea is if you check how the healthy cell looks like, they have 46 chromosomes. Each of them have 2 copies, and if we count, we have 0 copies of 0 chromosome with 1 copy, 23 chromosomes with 2 copies, 3 chromosomes with 3 copies, and so on. And actually, we can say, okay, maybe we can just describe our karyotype by these numbers. So we're introducing something like this. So this vector obviously substantially reduce the space. We can calculate now this, and we still have some important informations here. For instance, you can see that though this cell has 46 chromosomes, it is not now the healthy diploid cell because it has 1 copy of chromosome 1 and 1 copy of chromosome 3, and altogether we can see that this karyotype is different from this one, and also just by observing how this looks like, it's quite obvious that this is much more diverse, but also in this macrokaryotype representation, it's already present there. Thanks to this, we can now start developing our theoretical model in which we aim to go through many generations and to follow what happens during multiple cell divisions. Our aim was to describe a larger number of cells, and larger number of cells means going there by a mean field approach where number of cells is described by this n, and this is simple rate equation where division is described by this term. Then in addition to successful division, some cells can actually undergo apoptosis or cell death, or cells can also misaggregate, and for all these 3 reasons, actually they somehow contribute to how the number of cells changes there. Of course, to make the model complete, we also need to take into account that this type of gain terms appear where when this type of cells misaggregate, it can actually lead to our original cell karyotype. So they are also contributing with a positive contribution here. So this is basic idea of the model. All these parameters need to be actually carefully calculated they're not as simple as they look at first glance. Of course, we also wanted to now study what happens in this in real system. This is experiments that are done in COPS lab by Thomas, and the idea of these experiments is they from patients they extract these pre-tumor cells, and from these pre-tumor cells they evolve them in vitro. What that means, they extract these cells, grow them on the, in an artificial surrounding, so they put it just on the cover slip and they grow there, and when they grow enough, they can take the cells from there, from this cover slip, and analyze the karyotype sequence there. Also, they can keep a certain portion of these cells and grow them again. That's something what sets one passage. And they do this in a controlled manner, so they know exactly how the number of cells that are keeping during each passage, so they each time are getting 1/6 of the cell initially at the certain passage. So this is about the method they are using. Now, I know that this is maybe not so familiar to everyone here, so let me explain what they are doing afterwards. They are taking cells from their population, and they are doing single cell sequencing. What means single cell sequencing? They can actually search for the karyotype of one cell, and here are roughly 50 cells shown with colors here denoting the number of copies. Green means 2 copies, so all these chromosomes are 2 copies. This one is red, means 3 copies, then green again 2 copies, and the last one has 1 copy. Of course, this is how it looks like one passage, but let's now check what happens with when they are doing these passages. What they found actually is they have like 2 types of this evolution that they can observe. And first I will explain, I will introduce this one that is more simple and easier for understanding. So pay attention to the chromosome that are here labeled. This is chromosome 18. You can see how in time the number of cells having 3 copies of this chromosome change and grow. If we plot these numbers in time, we are getting this type of change of this chromosome. So from almost 0, it's going to almost 100%. We wanted to understand and explain this type of behavior, and we didn't do first our full model. We just wanted to have as simple as possible model, and actually we can do it with the extremely simplified version of the model, where all these karyotypes I'm not showing. I'm showing only 2 types of karyotypes, one with 2 copies of certain chromosome and 3 copies of certain chromosomes. And this is the rate equation that describes what happens with these copy numbers. The nice part is of this model, we can actually calculate this analytically. This is analytical solution, and this analytical solution can be also fitted to the experimental data. From this fit, we can say something about cell lifetime, and we can say about misaggregation rates, and we can say what happens with the cell lifetime for different chromosomes. The main conclusion of this calculation is that cells that are having this gain are actually proliferating faster, they're dividing faster than the cells that do not have this gain of chromosome 18. The other fitting parameter was this misaggregation rate, and the model wasn't too sensitive to this number, so it is pretty much convincing that the main driving mechanism relies on this faster proliferation chromosome 18. Now we took these numbers and put them in the full model to see whether we can explain what happens there. And there is no difference in this behavior. Again, we can explain how this chromosome 18 is going from 0 to almost 100. But here you can see some discrepancies. That chromosome 20 here has the same, so almost all cells have 3 copies of chromosome 20, whereas if you check this red line, it suggests that it should go down. So it was obvious that in addition to the parameters that I have here, I also needed some additional information. So for other karyotypes, we said that they proliferate slower than the original karyotype and also than the karyotype of this chromosome 18. When we did this, actually we could explain the observed karyotypes completely. Please note that what we said here, it is huge parameter space, and because of this huge parameter space, we couldn't say that all other karyotypes should proliferate at this rate, but definitely the neighboring karyotypes should proliferate that fast to explain what happens there. This is something what we do like is our prediction tells us what is the times between these passages. It drops, so blue line is theory and red dots are experiments. This suggests that indeed in experiments and in theory they proliferate faster. Yes?
Nenad, what's the axis? What's the y-axis there? That's days.
Days.
And can they only proliferate once per, on a daily basis, or
yes.
why is it discrete there?
What is the?
Why is it discrete?
Why is discrete?
Yeah.
Because of the method. Because of the method, they do passages once per day. So they grow these cells, and when they grow enough, then they do passages, and they just consider that the moment when actually they did.
They make a measurement every one day.
Yes, that's an inaccuracy in the measurement. Yes. And here is if we want to simulate, because it's always nice to show that something what is so robust in mean field approach that is also robust in stochastic simulations. So here we have stochastic simulations, and again we can see that indeed there is a small amount of these chromosomes that are appearing here as some deviating from these 2 copies, and here we have that actually we are getting 18 and 20 with 3 copies. What this model couldn't completely rule out is if this is about proliferation or is about cell death. And we also try to explain this behavior by setting the parameters that are having a different rate of apoptosis, and we could again explain this behavior by a cell death. So apoptosis or cell death can also explain the data. So our conclusion is that either is proliferation or apoptosis, or even interplay between these 2 processes. Now I would like to go back to experiments and to see what happens there. Because I already initially mentioned that we have the 2 mainly two different types of karyotypes appearing there. And this is something for when they did sequencing from a different patient. So before was one patient, and this is the other patient. And here you can see a dramatically different initial karyotype. Here, I would like now to analyze this data also by, not only by doing this, by showing this, the way how they represent the sequencing data by these colors, but also I would like to show you the, how this data look like in the macrokaryotype representation. And you can see them, they are mainly here in this cloud. They are grouped here. And what does that mean? In this karyotype representation, this corner represents the karyotype with 2 copies, here with 3 copies, and here is mainly with 4 copies. And what does this mean? That if we are close to this diagonal, this means that we are close to the having either 2 or 3 copies. Of course, if we now check what happens in time, so how this karyotype evolves, you can see that this cloud moves now in this direction. And if you go more and more in time, you can nicely see that it converges somewhere towards near diploid state. We wanted to understand this behavior, to explain it, and what you will see that we can guess something, but we don't have entire picture yet what's going on there. So what we wanted to understand, so we wanted to somehow systematically approach to this problem by starting from the most simple idea. So what Lucia did, she started from this cell and evolved this cell with the very same parameters for all karyotypes. And what happens if she was doing this, she could see that mainly for relatively small miscirculation rates, it moves not along this diagonal line, but away from this diagonal. And for larger miscirculation rates, it moves much away from this diagonal, which means that miscirculations and the constant coefficients cannot explain here. So there is a certain selection in this system. Then we played a bit more with parameters. So we penalized cells with 1 copy and with 5 copies. When we did this, then we could see we are getting now staying closer to this line. But it wasn't good enough for us because if you remember, so just compare this result here and this result, and at first glance it's obvious that we are far from the explanation. So what we wanted to do, we now started to put some additional advantages on the cells that are having these 2 copies. Of course, we put something by hand, and we could see that we are getting closer to the karyotype that appears in experiments, and they are almost now getting close to each other, though there is a certain, this one is more heterogeneous compared to this one. We wanted to make it even more homogeneous, and to make it more homogeneous, we played with miscirculations. What we said here, we say that miscirculation rates changes as we are going from this far or strong aneuploids towards near diploid states or near triploid, whatever. But if we are going in this direction, miscirculations will not be that strong as here. Of course, the result was much better, and this is close to what they see in experiments. So we know something about this interplay between the misaggregations and some other things that are doing the certain selection to go in this direction. Here is the simulation, and if you pay attention to this simulation, you will see that we are still not close enough to experiments, because on this road from this initial karyotype towards these final karyotypes, there was a certain transient point where actually we had a lot of these heterogeneous states. So this is a real challenge, and we are discussing what else could be there. I don't have the answer yet. Okay, and now I'm going back to experiments to show you also something very dramatic, what happens there. There are experiments that have this transition from something what is close to the diploid, near diploid states, towards something very, very, very diverse, some really dramatic change. And the question was, can we explain this transition? This is something what they do see in experiments, but also we know that something like this can happen during evolution, because we quite often can see similar karyotypes, and we know that our cells are diploid, so we have quite often this type of transition during tumor karyotype evolution. And the idea was to check whether it's going via multipolar spindle, or it is related to whole genome duplication. So how we model multipolar spindles, we say that we have, for instance, 3 poles, and if we have 3 poles, we have the same probability for each of these connections. And then, of course, if there is this type of cell division where 2 poles will end in one cell, we will get certain number of chromosomes there. And this is what we are getting just by calculating probability of these macrokaryotypes. And what is nice here, that if we compare this to the experiments that I have shown before when I was discussing this transition from this point to this point, this cloud and this cloud are pretty close to each other. And this is the other experiment where the cells are also somewhere here. So they are also pretty much close to this cloud. So this suggests that multipolar spindle can explain this dramatic change, this dramatic transition from diploid states to these really aneuploid states. Alternatively, this can go in via different pathway where the cells, it is known that cells can duplicate their chromosome content. If they duplicate their chromosome content, then they are also, after having 4 copies of chromosome, they are actually in a really large percentage dividing via multipolar spindles. So again, something similar like this, but just in 2 steps. So here I'm reaching my conclusions. So I have shown the reason why we need and why we are so excited about this macrokaryotype-based model. I have shown that we can explain this transition during this tumor evolution, and also introduce why this multipolar spindle can actually be a plausible explanation of what one see in experiments. Here, I would like to thank first Lucia. He is somewhere here. Hi. And also I would like to thank Ivan Naban, working on this macrokaryotype model. Then I would like to thank Kops and Thomas for this NIX experiment, Eva first for long-term collaboration, also helping us with this, building this model, and Mariana Tracala for cells that are help us to actually understand what happens in some other system. So here I would like to thank for your attention and take the questions.
Hi, thanks for the talk, sir. I have a question about how the single cell sequencing, the data was interpreted. So after each passage, I could see that the vertical columns had different colors. So how the copy number was decided?
I just would like to some of these examples, like this one.
Yes, like in the vertical bars,
Yes.
there are different colors, and each color was correlated with a different copy number. So
For instance, this is 4 copies, and this is 3 copies.
yes, so in
What do you mean by interpreted?
like for each chromosome, what number did you take?
For all these numbers,
Okay, all these numbers
all
were
these
taken.
numbers are taken, and we are just, if we want to show what happens somewhere, like in these cases. Wait a second. So these are experimental data. Each point here represents one cell.
Okay, okay.
So this is exactly all these cells. For each of them, we place them somewhere here in this graph.
Okay, I understand. Thank you.
Yeah, question. Thanks. So how often does this happen, this multipolar error? Has this been estimated experimentally? Do you know?
Depends really strongly on which system we are discussing. If we are in tumor cells, they are quite often. They are quite often when there is multiple number of these centrosomes. Because what sets the number of poles is to a large extent also by centrosomes. And in normal healthy cells, cells have 2 centrosomes, and they are just nucleating 2 asters, and they are forming a bipolar spindle. If cells undergo tetraploidization, means it is completely duplicated, it also duplicates number of centrosomes, and it ends with 4 centrosomes.
Yeah.
In that case, cells are now starting asters. Mainly they can even form 4 asters, but typically they are forming 3 asters. It's not known why they are preferring 3 more than 4, but also cells with more than 3 asters are observed. So,
Thanks. Yeah, and maybe another question. So, so is this indeed strongly correlated, the cell division time, with this missegregation? You briefly, I think, discussed this, but is this generally true that that the cell division time correlates with missegregation, for example?
in the model, we don't have correlation between misaggregations and cell division time. All these parameters are related to the chromosome content. So if the cell has a certain combination of chromosomes, it can have different proliferation rate. So it can divide faster or slower, because it's quite obvious that cells carrying different number of genes, then they will not divide equally fast. So that's our assumption.
Yeah, okay, I understand your point. Yes, thank you.
Hey, so this dramatic change that you showed, in certain cases there is a huge deluge, and then before it can go back to diploid. This is very reminiscent of this directed percolation transition. Because I think in the earlier part you had showed that the chromosomes could be, their rates of change could be affected by nearby chromosomes. So do you think this sort of, the
Can you
transition?
repeat again the question? I didn't get your question, sorry.
So this dramatic change
Yes.
is sort of reminiscent of directed percolation transition, where if you change a particular rate, then you see a dramatic increase in the number of active states. Depending on the model, you can define what is an active state. Here, these in the passage 20, all of those are mostly active states.
Okay, one can think about this as just pure stochastic process where you have higher, lower misaggregations. But this is something what is not known, that in cells there is a huge misaggregation rate in cells that have just normal bipolar spindles. I'm not aware of that, something like that. Whereas in cells that are forming these multipolar spindles, they indeed can have really a huge misaggregation events during a single cell division. And this is this is biologically very different situation, and I think it will be, I mean, there is a huge debate in the field what causes this transition. So it's not, I mean, I don't know how many labs are trying to answer this. It's not simple question.
Nenad, what cells are these that you're, what's the cell type that you're looking at here? What cells are these?
This is adenoma.
It's a tissue culture cell.
Yes, solid.
And is it
solid.
is it an organoid, or is it?
Yes, this is on organoids. So this is not it's not just a colony. This is organoid.
So an
experiment.
organoid is growing,
Yes.
and then you're taking samples of the growing organoid.
Yes, that's sample
And
of organoid.
you can take a certain fraction of the cells out of the
Yes.
organoid, and then you
Yes.
do it. So that means that there's a lot of different cell types in the organoid.
It could be that also influences the heterogeneity of the system. Yes.
And what would happen if you were looking at it in a tissue culture cell, for example, if you did the same, not an organoid, but in a tissue culture
I don't
cell?
know the answer.
Okay.
I don't know the answer. That's yeah.
No, I mean tissue culture. No, sorry, I mean
But that's-
isolated. A cell line.
Yes.
What if you did this in a cell line, not in an organoid?
I mean, here, really, as Anatoly mentioned, they grow and grow and grow. They just grow. And when we passage them, we grow them again. And I don't know if one would keep all these cells, to which number eventually we'll get. But it would be much, much more than the number of cells in our body.
Do you have predictability now if you were to do karyotyping of cancer cells, of already matured cancer cells? Can you predict the path that cells would have taken to get to that endpoint?
That's exactly the aim of this modeling. And we can say something about this. So one thing what is clear about this path is that we need to say what happens with misaggregations. We need to start with higher misaggregation and then go towards lower misaggregations. That's one thing what we learn from this modeling. And the other thing what we learn from the modeling, there is a strong selection here. It's not easy to explain. It's not easy to explain this. So here, just you can check how homogeneous are these patterns here. Like the cells in this upper part, they are almost identical. Whereas here in our theory, it's not that the case. So we are not yet close to this experimental point. So, but to get closer to experimental point, we need to set some some selection, and this selection is on the, for instance, proliferation of cells with more copy numbers, like 5 copy numbers, 1 copy number.
Yeah, or even death. I don't know if cell death is cell death. Is that
cell death is is kind interchangeable, and by the theory we cannot say whether it's proliferation or cell death. I would say mainly it's about the proliferation, but theory cannot make the difference. I think only experiments can finally say what's going on there.
Okay.
Thank you very much. I'm afraid it's a question half for you and half for the previous speaker, but I guess you're both involved in the same thing. So is there a role for the flexibility, the bending flexibility in the microtubules in the formation both of the more than bipolar structures, because they have to flex less if you form like tripolar, and then also related with the bending that was described in the previous.
Okay. First, related to the first part of to the first question, I'm not aware of any study of this theoretical study of mechanics of these multipolar spindles. We are now actually our next step will be trying to understand from theoretical perspective multipolar spindles, and as you mentioned, I think there is a certain reason for this to believe that actually this flexibility of microtubules, this bending rigidity of microtubules will also set more than two poles. Related to the about the second question, it was about the pivoting and how how these microtubules pivot. Mainly, it is a freely joined, this link between the pole and microtubule is freely joined connection. So if your microtubule changes the angle, if there is no force that will revert back to some other initial point, it will stay at the new direction. So once when you reorient, it stays there. If here this link would be some kind of elastic, not freely joined, then actually because just of this elasticity, it will be rotated back. Experimentally, it's not observed this reorientation back to the original orientation.
Okay. Just let me give you a context. I mean, and there are many mutations known in tubulins that are the proteins that made up. I mean, just for eventually. And in most of the cases, you know that those mutations fall into a protein-protein interface, so it's kind of trivial to say, okay, this affects energies or whatever. But many of other mutations that are more cryptic are in places that are exterior to the microtubules, and nobody knows why they affect, or even it's very difficult to know what they are doing because there are apparently no interactions or stuff like that. So some of the ideas is that they either affect the speed of growing or melting, say, of the microtubules. But now the question is, you said that this moves like this way, but what if it can more, I mean, flex and be more elastic and reach more to the central part where the chromosomes are divided? Would that be possible?
So the flexibility plays a role in this setting the shape of the spindle. The shape of the spindle shows these microtubules that are bended. And it's quite obvious that there are different experiments showing that these microtubules are indeed bended. If you do laser cutting experiments, it will get straight and it will move in a certain direction. So microtubules are indeed bent when they're in the spindle. But the microtubules that are pointing not towards the spindle, towards the central spindle, but if they are pointing towards the cell cortex, they're straight. So, was that the question?
Would it be possible to include cell competition in this? Because I imagine from the model that would be quite difficult to treat, but the same dynamics could in principle emerge from I mean, if you had each karyotype is equally viable, but when put together they compete in a way that is...
What would be cell competition? I don't understand
you would have
what you mean by that.
A composition of different karyotypes which interact in a way that is not...
Like neighboring cells interact?
Yeah, population, I guess.
I mean, there are studies that actually also, experimental studies, that suggesting that cells can influence each other. But so far, I would like to understand what happens here on this, I would say, pretty synthetic system, because it's a very simple mathematical approach, rather than going in that additional complications. I would like to see whether we can explain what happens in this system just by this model. I think we are not too far from explanation, but I'm not for introducing additional unknowns in the system.
But in the system, the population is heterogeneous, right? In one organoid.
I think by definition of organoid, it's not homogeneous, because
Right.
organoid
In
does
terms
not
of
grow
karyotypes.
just as colony, because they have a certain hierarchy how it's growing. I mean, Eva can tell you better, ask better this question. I'm not expert in biology.
Thanks.
Let's stop now and thank Nenad and all the speakers of this morning session.